Welcome to CSEC Biology, the cover page. I am Mr. Wilson, and today we're going to be looking at the properties of enzymes. In our previous lesson, we looked at what are enzymes. Just a reminder, enzymes are biological catalysts which actually speeds up the rate of a reaction. Important to note is that enzymes are made in the cell, and of course, they are all protein. And here we have the elements that protein is made of: carbon, hydrogen, oxygen, nitrogen, sulfur, and of course phosphorus. And the cell there, just a place where the enzymes are made. The first property of an enzyme is that enzymes are specific. Here we have carbohydrates acting on starch, and carbohydrates will never act on protein. Likewise, protease will never act on starch. Now, one of the reasons for that is that enzymes have what we call active site, and here you're seeing an enzyme and of course the active site. Now, the enzyme here will only of course act on this type of substrate, because this type of substrate will nicely connect to the active site of the enzyme. So we see here that enzymes act on only one type of substrate. They are specific in their action, and that is of course one property of enzyme. Let's look at another. The second thing is that enzyme, their chief function is of course for chemical digestion, and of course they break down large bonds into smaller units. Look at the example here. We're breaking down starch, and of course amylase is breaking those big bonds of starch into smaller bonds called maltose, and of course maltose is acted on by another enzyme called maltase to eventually break it down into glucose, which of course the body will be using as a monosaccharide. So here we say for 2, this is the second property: enzyme breaks down large bonds into of course smaller unit. Now, the third thing we want to know, of course, a third property of enzyme, is that enzymes are not chemically changed at the end of a reaction. And you'll remember our enzyme from our first slide looks pretty much like this with the active side, and it is of course acting on the substrate as seen on screen. So here we look at the enzyme combined with the substrate. Of course, the bond is broken there, and you will realize over here the enzyme will of course release the product, and there the product is. But look at the enzyme remain pretty much the same. The enzyme is of course chemically unchanged at the end of the reaction. So there we have the third property of enzymes, and we move on to the fourth property. Enzymes work best at a particular pH, and we refer to this pH as the optimum pH. While the enzymes might be working in a pretty much a range, as for salivary amylase will work at a pH of about 7 to 8, and pepsin pH of 1 to 3, and trypsin, which is of course found in the small intestine, will work at about 7 to 8. But if you look at the graph over here, you're nicely seeing it intercepted in the middle there, and it shows pretty much the optimum pH, and that's pretty much the best pH, or where the enzymes will perform at their best. So the fourth thing here, just a reminder, enzymes are at best at a particular pH. You want to ensure that you're following the lesson, because you're going to be having a little quiz at the end of the lesson. And we'll move on to 5. Enzymes work best at an optimum temperature, and that's about 37 degrees Celsius. That is pretty much the human body temperature, somewhere around there. And of course you're seeing the graph again showing of course temperature and of course the rate of reaction, and you'll realize there that it is of course intercepted right at the point of optimum temperature. So here we move again. Enzymes work best at a specific temperature, or around a specific temperature called the optimum temperature. For human, it's about 37 degrees Celsius. Interesting here, enzymes are denatured by high temperature. It's important to note that enzymes are not killed, because enzymes are not organisms. They are actually chemical. You would have seen this in our first video on enzymes. So enzymes are denatured by high temperature. There we have the fire. So if you were supposed to go too close to too much fire for too long, then you understand that the enzymes will be denatured in you, and of course eventually you'll die. Here we have a leaf, and it is a leaf that is propagated asexually, but it has been exposed to hot water. There you're seeing it in the beaker with the hot water. Now the temperature here is way too high for this leaf. What is going to happen is that, of course, the enzymes in the leaf will be denatured, so of course this leaf will not be able to grow at the end of this experiment. And again, you're seeing the graph there showing that if, of course, we pass over a certain temperature, then the rate of reaction starts to fall. And of course, you know, eventually the organism will die. And we move on to the seventh property of enzymes. Here we go. The seventh property of enzymes is that enzymes are denatured by some poison, example cyanide and of course arsenic. And of course denatured won't work, you'll die. And here we have a skull that we got from our friends over there at Pexels. To access the skull, you'll find a link in the description below. I hope that you enjoyed the presentation. Please be reminded to like, share, and of course subscribe, and check out our many videos that are posted weekly. We also have 3 weekly live classes on science on a Wednesday, Friday, and of course Sundays at 5:05 p.m. Let's look at what's in store for us. How much do you remember? Alright, so we have our first question here. Dash or biological catalysts that speed up the rate of a reaction. Let us see what you have here for us. 5 seconds to answer. I'm sure you're a bright chap. Let me see what you've got. Let's check the answer. Wow, that was wonderful. Of course, enzymes are biological catalysts that speed up the rate of a reaction. Let's look at the true and false binomial question. Enzymes are small organisms that speed up the rate of a reaction. Is that true or false? Ensure that you read the question carefully. Enzymes are small organisms that speed up a reaction. Is that true? Let us check. Oh no, it is not. Why is it not? Enzymes are not organisms. It's actually a chemical, not of course a organism. So enzymes are pretty much chemical, and they speed up reaction, but they are not organisms, which makes this false. Question number 3. The substrate does not determine the enzyme that will act on it. What's your answer? Let's check it out. Of course, it's false. Enzymes do determine the substrate that they will act on, or the substrate determine the enzyme, as enzymes are specific in their action and will only work on a particular substrate. Let's look at the other question. The higher the temperature, the better the enzyme will work. Example, about 7 to 8 thereabout. Is this true or false? 7 to 8 degrees Celsius. Let's check it out. It's false. 7 to 8 here would have been the pH for the amylase and of course for trypsin, not of course the temperature that the enzyme would be working at. So here we have higher temperature. No. Enzymes will work best at a particular temperature that we refer to as the optimum temperature. True or false? All enzymes will work best at optimum pH. I guess we all have this one. It's true. Yes. All enzymes will be killed by poison. All enzymes will be killed by poisons. Or false? Enzymes can't be killed because they are not alive. Alright? it's actually chemical, so of course you can't kill the enzyme. You can actually denature the enzyme. Thanks much for watching. You've been listening to CSEC Biology, the cover page. I am Mr. Wilson. Please be reminded to like, share, and of course subscribe. When you subscribe, ensure that you hit that notification bell and of course select all so that you will be notified as soon as we have a publication. Thanks much for watching.